Hello, grade 10. Today our lesson is section 4 in chapter 9, genetic engineering. Our key concept today, DNA sequences of organisms can be changed. Let's watch together this video about genetic engineering.
So, what is genetic engineering? Genetic engineering is when the genetic code of an organism is altered to produce a desired trait. This is done by taking a gene from one and inserting it into the DNA of another to get the wanted end result. To get a better sense of how this is done, let's first understand how DNA works. In any living thing, the DNA acts as the instruction manual for the body. Certain genes code for certain proteins, which in turn make up all the different parts of the organism.
So, genetic engineering, as we said, is changing the DNA sequence or certain gene in an organism, okay? Altering this sequence. Why? To produce an organism with desired trait, with better traits, okay? And as we learned before, the DNA is made of genes. These genes which code for protein synthesis, which in turn related to the color, shape, and all the traits that you have or an organisms have.
Any trait that you can think of is coded by DNA. Now, once you know what codes for what, you can use genetic engineering to essentially edit the parts you want, to some extent. All living things use the same genetic code, DNA, made from the same 4 bases, so scientists can use special enzymes to cut parts of the DNA from one living thing and sort of sew them onto the DNA of the target. The result is then a living thing that has a new gene and a new feature as a result of that gene. Now, there are many examples where genetic engineering is used today, but we're going to focus on a very specific medical example to understand this concept better. Humulin, a medication used for diabetics, is created with genetically modified bacteria. Humulin can be given to diabetic patients who lack the insulin they need.
So they use genetic engineering in medicines, and humulin is one medicine which is needed for the diabetic people. And as we know, the diabetic people, they lack insulin, okay, because the pancreas is not able to produce insulin. So they will have high glucose levels in the blood. They cannot enter the cells. So this medicine was produced by using bacteria by the genetic engineering. How this happens, let's see.
Now how they do this is scientists figured out which section of our DNA codes for insulin, and they also found certain enzymes that can cut that gene out.
So they found out which gene, which sequence on the DNA of a human codes for making insulin. And they use specific enzymes to cut this part. They use this part and insert it in a plasmid of bacteria.
What they do is, using enzymes and heat, they isolate specific gene and then insert it into the DNA of bacteria. The result is now a bacteria.
This is a DNA of bacteria called the plasmid. Okay, this plasmid is DNA part in the bacteria. Now this plasmid will have this gene, okay, which is able to produce the insulin.
Bacteria that produces human insulin. And since bacteria can multiply really fast, you can create a lot of insulin.
So the bacteria will be cultured and they will multiply very fast, reproduce very fast asexually. Then they can collect the insulin produced by the bacteria and use for humans
in a very short amount of time. Now, this is great. Genetic engineering poses huge potential in the field of science, providing us with new insight on how our genes work. However, there's also a lot of controversy in certain respects to genetic modification, specifically genetically modifying our food. To learn more about GMO foods, check out our next video.
Okay, there are disadvantages and advantages for the genetic engineering, which means genetically modifying, changing in the DNA sequence. Okay, there is one thing also in in genetic engineering which is called cloning, okay? And a clone is a genetically identical copy of a gene or of an organism, okay? The first cloned animal was Dolly's sheep, okay? First of all, we just should know that cloning occurs in nature through asexual reproduction. That means some organisms like bacteria, they can clone themselves by nature because they divide by binary fission to produce new cells. And some plants also, they reproduce asexually from their roots. As well as any simple animals like the yeast and the hydra, they can be reproduced by budding asexually. Okay, and this is type of cloning that happen naturally. But mammals can be cloned through a process called nuclear transfer. They cannot be cloned naturally because mammals reproduce by sexual reproduction. This process happens by using from the name of this process, nuclear transfer, that means they transfer a nucleus from one cell to another, okay? So the technique or the procedure for this process is they first take a nucleus from an egg cell. They take an egg cell from a sheep, okay? Now they want to make a exact copy of Dolly's sheep, okay? They take a body cell from Dolly, all right? They take out the nucleus, which has all the DNA set, the genome of Dolly. They take the nucleus and they insert it in an egg of another sheep, okay? So nucleus is removed from an egg cell and replaced, okay, with the DNA or the nucleus of Dolly's sheep. So nucleus of a cell from an animal to be cloned is implanted in the egg. After this, this egg will be inserted in another female sheep and it will start to divide, okay, and produce the new offspring. Now, as you see here in the figure, egg cell, they take out the nucleus from the egg cell. It's extracted and discarded. We don't need it, okay? And here, this is a body cell. They take the DNA from the body cell, اللي هو nucleus, and they insert it in the egg cell, as you see here. The DNA from the body cell is extracted and, okay, and, sorry, okay, the DNA from the body cell is extracted to be used and the rest is discarded. They just only take the nucleus and they put it here. This will start dividing, okay, stimulated to divide. After a few days, they will implant it in another sheep and the egg will keep on dividing until it becomes a whole new organism or new offspring. And this organism will be exact copy of the DNA that cloned here. Okay, we will stop here today. See you tomorrow and take care, guys.